Okay, here I am. Hello again, everyone. My apologies, I will be speaking in English. I do know a few languages, but French is not one of them. Okay. Yes. As I mentioned, I come to you from Deakin University, and in Deakin University, in Food and Mood Center, we pioneer research in nutritional psychiatry. So Professor Felice Jacka presented to you yesterday and discussed what nutritional psychiatry is. It's the area of research and practice which uses diet as one of the key modifiable factors which are able to support mental health. We, of course, understand that our mental health is impacted by many determinants. There's genetics, environmental, psychosocial factors. However, we now understand, recognize, and have evidence to use lifestyle medicine and lifestyle factors such as diet, exercise, sleep, stress management in supporting mental health care. And it is quite important because we understand that in the last few decades, the burden of mental illness continues to increase, despite our best effort, despite new research being done in the area. We understand that pharmacological approaches proven to be insufficient to address this burden. Also, of course, pharmacological approaches, although needed and essential, can't really address comorbidities. We know that majority of the people living with mental health illnesses have at least one comorbid disorder. Most of them have 2, 3, 4. Usually, it's metabolic disorders. We understand that adjunctive dietary treatment is a successful strategy for significant symptom improvement. It also supports physical health, it supports behavior change, and it's happened to be highly cost-effective. Most of the studies I'll talk to you about here done cost analysis and showed that using dietary approach in treatment of mental health conditions is actually cost-effective. So let's have a look at a couple dietary interventions. First of all, of course, dietary interventions being used in support of mental health care and people living with mental health illnesses for a few decades now. There's various approaches used in dietary interventions: individual, group, mix, in person. During, of course, lockdowns, the latest COVID lockdowns, we moved online. In Australia, or where we live in Melbourne, we endured some of the longest lockdown periods in the world. So we used a lot of online counseling. There's also various dietary styles. I'll talk a little bit more about Mediterranean style, but it doesn't have to be Mediterranean. It could be anything based on guidelines, or healthy, or ketogenic, or anything which have evidence behind. Usually when we use dietary interventions, the length is about from 4 weeks to 12 months. Here I need to tell you that, of course, the longer you have, the better chances you have to change behavior. And when we talk about outcome measurements, majority of dietary interventions use metabolic health as outcome measurement, and only limited number of studies used mental health outcomes, such as depression measures or quality of life. So this systematic review we published a couple years ago, and it's specifically explored the effectiveness of nutrition and dietary intervention for people with serious mental illness, and when these interventions address physical health. So there's a QR code for you to scan should you wish to read more. It's a free publication online. So what we found that outcomes are quite inconsistent, but it's still not to discourage you and to say that there's no evidence in using dietary interventions. We, for example, found that when interventions were designed and delivered by dietitians, it's actually improved effectiveness. And that's what we see consistently through the literature. So when you design dietary interventions, you do need to involve professionals. As I mentioned, there were few interventions which specifically focused on mental health as primary outcomes, most of them being done in people living with clinical depression. That first trial, the SMILES trial, came to you from a Food and Mood Center, and it was first randomized controlled trial which actually used diet to treat mental health symptoms. After that, we had trials in young males. That was a very interesting, that bias trial here. Very interesting online study, and in young males, you know how challenging young males usually are to treat and support, and that was actually very, very successful trial as well. Sorry. We, of course, have Healthy Med trial, which is very similar to the SMILES trial. It's used Mediterranean-style diet which, with fish supplements. We also have very brief trial in university students, which lasted 3 weeks and also was quite effective to help some people to improve their mental health symptoms. And also the CALM non-inferiority trial. That's a trial which our center ran online, and it's actually showed pretty good outcomes, but we haven't published them just yet, so I can't talk about them. What I will talk about is the SMILES trial, the very first randomized controlled trial which used dietary intervention to support mental health. So we recruited 67 adults living with clinical depression. So it's not just people who answered questionnaires and showed some distress. That's people who diagnosed with clinical depression, and some moderate to severe. And a lot of them lived with that condition for a while by the time we enrolled them in a trial. So we split them into 2 arms. One arm went through dietary intervention, and another arm went through befriending protocol. It's lasted for 12 weeks. So in dietary arm, we used Mediterranean-style diet. So it was a combination of traditional Mediterranean diet and Australian guidelines for healthy eating. Because we ran the trial in Australia, we tried to adapt the diet to be most kind of suitable for the Australians. So the main principles of the SMILES trial diet was individualized approach. So that diet was designed by a dietitian and was delivered by a dietitian as well. So we did use person-centered recommendations because dietitians do have enough training to understand clinical needs, but also nutritional needs of the patients based on their knowledge, skills, resources, social environment, and many other factors which can, in fact, impact our eating habits. So it was all about behavior change. So dietitians help participants to set up goals to change their behavior, to follow up, to revise, and they provided consistency and support. As I mentioned before, it's lasted for 12 weeks, and this is an example of the pyramid of modified Mediterranean diet, which was used in a trial. As you can see, very similarly to Mediterranean-style diet or Mediterranean diet, it was based mainly, It was based mainly on plant-based foods. So lots of grains, around 5 to 8 servings, and of course, because dietitians had opportunity to sit down with every patient, they could adjust the number of serves based on the needs. We also recommended for people to eat lots of vegetables, 6 serves of vegetables, lots of fruit. Dairy, olive oil was recommended as main fat. In fact, we recommended for people to use up to 60 milliliters of extra virgin olive oil. We also recommended for people to consume nuts every day, legumes and beans a few times a week, red meat, which is usually a very rare occasion in Mediterranean diet, but because we were working with Australians who love their red meat, we increased the amount here. and all other food groups according to Mediterranean diet. What was, of course, interesting and in accordance with Mediterranean diet, we asked people to decrease consumption of so-called extras. So those ultra-processed foods with high addition of sugar and salt and fat. And we asked people to consume about 3 serves of this a week. When we enrolled people into study, they on average consume, I think, 5 to 7 serves per day. So that probably was the biggest change. Because I'm trying to show how to actually implement the recommendations in practice, I'll just go through the kind of, session plan here with you. So we had 7 sessions in the SMILES trial, which were stretched over 3 months. So session 1 had 2 broad objectives: to assess current situation, remember they were one-on-one sessions, to figure out motivation, intake, needs, resources, skills, and to cover the basics, to provide just enough information so when people leave, they actually know what to do. Not to overwhelm them with information, but to provide just enough. Sessions 2 and 4 started with reviewing the goals and how people went, with discussing specific concerns. We really do need to address concerns and not to dismiss them. Celebrating wins, that's very important. That's always good to know that people doing some changes and acknowledge this is very important. Encourage new goals, talk about strategies, talk about barriers and provided resources. And session 7 was about long-term plans and strategies. And we still have actually people writing to us and talking about how this trial actually helped them with their lifestyle. So 12 weeks and look what happened. Our social support group improved their depression score, but look what happened in diet intervention. So many people actually improved their depression score that after 12 weeks, 32% of dietary support group achieve remission criteria. After 12 weeks, that's people who lived for years with depression. Okay, now let's go through a couple tips about implementing diet in your practice, because although I showed you a Mediterranean style diet example, it doesn't have to be Mediterranean, and majority of us, even without dietary kind of tertiary training, would know how to kind of eat better and are able to help our patients or clients. So we try to go through few steps to kind of make sense how effective delivery to support change might look like. So we try to say that, first of all, timing is everything. We do need to make everyone the center of attention. We do need to encourage small steps to basically go long way and to change behavior long term. We do need to motivate. We need to believe ourselves in change, because quite often when people come to us, they're living with severe mental conditions, you think, well, diet, it's probably the last thing that person can change. But people can change, and people do change, and we need to believe in that. We need to help people break the barriers, and we also need to understand that it's a lifetime commitment. It's not a short intervention which might change something, and that's it. We need to commit for life. So let's unpack some of these principles. So when I say timing is everything, it's of course very important to work with symptoms and treatment dynamics. So if someone going through crisis, talking about diet probably not gonna work. We do need to discuss with wider team, particularly if we're not a leading clinician for the case. We do need to understand exactly what's happening with our patient. We need to consider timing, when to start to talk about, how to go, how long to go for, what to discuss, when to review, and we do need to be quite sensitive and ready to be flexible. So just because we decided to start talking with someone about diet next week, it's might or might not happen. Where do we start? If you don't have a dietetic training, it could be quite challenging and confusing to know how to even start talking about diet. It doesn't have to be something complex. You don't have to start with dietary assessment. You can just ask people, what do they usually eat? Do they eat with family or friends? Do they follow a special diet? Ask them what healthy diet looks like to them, because we're all very different. There's a lot of misconceptions and misinformation out there. So it's very important to actually ask people what they know. Asking about knowledge would also help you to assess where people are in their readiness to change. Because of course, we need to be willing to change, to start the change. If someone in that preconception stage, in the very first one, they're probably not ready to even talk about diet. And if you start, you might do more harm than good. So maybe it's a good time to talk about sport or sleeping or something like that. If people getting ready, if they contemplating change, that's where we should see as our opportunity to provide resources, to encourage change, to talk a little bit more about. And all other various stages in our readiness to change, it's important to assess, to understand how best to approach the change. When I say we need to make people center of attention, what do I actually mean? Well, we do need to engage patients in managing their care. We know that the biggest concern people who are living with mental health disorders, they actually say that then when they're not involved in their own management, that's when all goes basically not as well as we would like to. So engage patients in managing their care. Gather information through active listening. Basically listen to what people want. Ask the questions. Be aware, of course, of their non-verbal communication. Respond to patient and also their caregivers' needs. Sometimes, we know that people don't actually do anything about their own eating behavior. It might be someone who is living with them. And share the decision-making. So when I say small steps, long road, so we do need to use motivational interviewing. We need to inflict the change. We do usually like to use SMART goals or objectives, but I know some people don't like words, goals or objectives. So you can use different ways. You can say different words. You can use words such as ways, or let's discuss what we can do about eating, or something like that. We do need to help patients to set goals, because setting goals helps to change behavior. We do need to acknowledge immediate needs in these goals. So if someone maybe doesn't have a budget to buy food, we need to discuss that first before advising eating more vegetables. However, we also need to consider core values. So there's a lot of things to probe and to understand, so we can actually help people plan short and long-term approaches. SMART goals doesn't have to be something like, I will eat 6 serves of vegetables. For some people, SMART goals might be, I'll go into supermarket and walk through veggie section and see what's out there, what vegetables don't scare me, what I can basically cook. People can create their own goals, and you can help them with that. When I say we need to be authentic, remember how I said we need to believe in change. Quite often in mental disorder space, we have that diagnostic overshadowing when we think that people who live with severe mental disorders might not be able to address their diet or lifestyle or anything else. They can and they do and they want to. And that's what we hear from people. So we do need to believe and motivate. We need to believe that people can change, and we need to support that change through various approaches. For example, providing tools or supporting capacity building through skills. When I say that we need to break barriers, change is very challenging, and we need to acknowledge that. If we say, look, people can do that, you can do it as well, it's probably not going to be helpful to that particular individual. So we shouldn't dismiss concerns like, oh, you'll be okay, you'll learn how to cook. No, we need to actually discuss that. We need to consider, of course, various cultural differences. For example, I was running a fantastic workshop with Foundation FondaMental just a couple of days ago. And people asked me there, well, French people really like to eat cheese and chocolate. What do you say about that? And of course, I'll say, who am I to tell French people not to eat cheese and chocolate? So we do need to consider various cultural differences and discourage that all-or-nothing approach. If we tell people never to eat chocolate, it's not going to work. We also need to use real-life applicable examples. How I just tried a few sentences ago, but I don't think it's particularly work. Anyway, we do need to help people with their knowledge and skills, provide foundational information, never assume what people know or don't know, use visual tools, use different scenarios, discuss easy food options, and as I mentioned before a few times, support building skills around food. So visual tools could be as simple as this. And through Food and Mood Academy, we can help you create visual tools like this. This is some of the examples from the people from CCU, so it's a community unit for people living with schizophrenia in Australia, and that's a practical examples how they do it there, how they use diet and treatment. For example, here, it's a wonderful, I'm trying to use a cursor, but it's not working. Over here. It's a wonderful selection of meals, which patients actually cooking together with whole medical team. That's some vegetables they grow in here. This is information about diet and nutrition, which everyone can access, read, and kind of learn from. Okay, so when I say it's not goodbye, it's see you later, we need to understand that everything can't be done in one session. We need to be quite flexible, and some people actually benefit from start, stop, or redo approach. We do need to engage in long-term commitment because it is a lifestyle change, and we need to communicate about it with everyone, including the medical team. Quick word about eating disorders. We know that, of course, our society puts a lot of pressure around appearance and around body image, particularly when it's come to young patients. There's a lot of unrealistic expectations. There's also a lot of stigma around diet. Every time when someone says diet, people think about dieting or losing weight. So we need to be very, very careful, particularly when we discuss in diet with young patients, because we, of course, here kind of walking on thin ice with the risk of developing disordered eating. We do need to encourage, particularly our young patients, to understand that bodies, of course, exist in absolutely various shapes and sizes, and it's normal. We do need to try to avoid negative language around food, because if we tell people, you can't eat that, it's too much sugar, or you shouldn't eat this in your condition, we really can't talk like this about diet. So we do need to use a body-neutral approach. We tried that in schools in Australia. When we talk about diet, we talk about gut health. So we don't talk about body size altogether. And it works because it's very inclusive. It's non-stigmatizing approach. So basically, as a practitioner, there's quite a few skills you need to be able to implement nutrition or nutrition interventions in your practice. Of course, you need to have basic nutrition knowledge, but also counseling skills to understand how to support behavior change. You need to be able to increase motivation, to support capacity for change and self-management, and you need to collaborate. Best, of course, it's to refer to accredited practitioner, such as dietitian, but everybody else can do a little bit to support dietary interventions. If you would like to learn more, we run Introduction to Nutritional Psychiatry course. It's been endorsed by Royal Australian and New Zealand College of Psychiatry. I'm a lead chair there in the course. Professor Felice Jacka is one of our experts. It's online, it's 3 weeks, it gives you 20 hours of continuous professional development, and you can complete that within, say, 6 months. And we start very soon, so hopefully I will see you there. Thank you again for listening. Please remember that although diet is a key factor in support of mental health care, it's just one of the determinants. But we all can do a little bit better to support our patients in changing diet. Thank you very much, and this is all for me.
So thanks a lot, Marion, and it's really a great pleasure to be here. And I'm really happy to be able to present some part of the work that we're conducting in the Lab Nutri-Neuro in Bordeaux. So my team name is NutriMind, and we are mainly interested in understanding how nutrition can influence brain function. And I will talk today about polyunsaturated fatty acid and its potential role in mood and cognitive disorders. So as we heard yesterday and today, nutrition influences brain function and, of course, is a determinant for one of the determinants for mood and cognitive disorders. And it has been illustrated, the consumption of a Western pattern diet, but also obesity and diabetes is a strong risk factor for this mood and cognitive disorders, while the consumption of prudent diets, and we heard about the Mediterranean diet, but there are other prudent diets, such as the Nordic diet, are more protective and associated to less development of mood and cognitive disorders. So, of course, all these data are observational studies that have been done in large population, and it's a general assessment. And one question for us is, how does it work? So what is it linked to? Why some diets are bad for the brain or others are more good? And our hypothesis actually is that in the diet there are some molecules that could act directly into the brain and influence brain function and then influence brain. So we get interested in the so-called polyunsaturated fatty acid. So there are two big families of this PUFA. The first one here on the left is the omega-6 family, while here on the right is the omega-3 family. So these omega are very important because actually we cannot produce them. So our body is full of this PUFA, and I will try to explain you what happened, really, why our body is full of this PUFA. But what is important is that the precursor forms that are found in vegetables are what we call essential because we cannot produce them. So we need to have them through the diet. So this is a very important information. And the second thing that you have to understand is that those two forms, so the omega-6 and the omega-3, are found in very distinct food. And for example, the omega-6, the precursors that is called linoleic acid, is found in sunflower, corn, and peanut, while the omega-3, that is the alpha-linolenic acid, is more found in walnut, flaxseed, or rapeseed. And these precursors, once we eat them, are metabolized by enzymes, especially at the level of our liver. And those enzymes are desaturase and elongase. And actually, there is a competition because this is the same enzymes that metabolize the precursors. So if you eat more of omega-6 as compared to omega-6 as precursors, in the end, you will produce more of the derivatives coming from the omega-6 as compared to the omega-3. And the interesting part is that the long-chain form of this PUFA coming from omega-6 is arachidonic acid. I will focus on this one because this one is found in the brain. And actually, those forms can also be found in the food, very specific, like milk, poultry, eggs, and dairy food. while the long-chain form coming from the omega-3, such as EPA and DHA, and I guess that you heard about those forms, are also found in fat fish and some microalgae. And what the recommendation today is to have a mix of this form coming from the vegetables and also to have the form coming from the animal sources. And of course, as people are not eating exactly the same quantity of those precursors and also of the long chain, overall the population is disbalanced in the ratio between omega-3 and omega-6. What is important for this PUFA, and this is why also we get so interested in this form, is that they are highly present in the brain. And for example, 30% of the dry weight of the brain is lipid, as you know, and 30% are PUFA, with DHA representing 10 to 15% of those PUFA. And actually, it's not only a question of DHA, but also of arachidonic acid. So both forms are found in the brain, and both forms are very important. Another thing that is really important is that those forms aggregate in the brain during brain development. So as you can see here, before birth and after birth, the level of DHA and arachidonic acid is going to increase in the brain, and then it reaches a plateau. And of course, the amount of DHA and arachidonic acid that we will have in this growing brain depends on what is brought to the baby, so through the mother, and it depends on the nutrition of the mother. Even if the mother doesn't eat enough of this, she provides it to the baby so she can get depleted, and actually it has been associated also to more depression during gestation. And once we reach the plateau, then the nutrition will allow to maintain this plateau, and this plateau will decrease also with age, and again the nutrition is very important to avoid this decrease with aging. And what has been shown is that DHA and arachidonic acid are very important for neurite outgrowth and synaptogenesis, and play a great role in this system, in the brain development. And what is known also, as I said before, is that because we don't eat enough omega-3, there is a global omega-3 depletion. So here, this is a map that has been established by the group of Clements in 2016, and actually they use a ratio of EPA plus DHA in the red blood cells. So this is a meta-analysis, and they show that this level is very low in some countries, and not surprisingly in the United States, but also in other countries, even in Europe. And while the adequate ratio, that should be close to 8%, is reached in only countries eating a lot of DHA of an EPA in form of fish. So overall, the global population doesn't have enough omega-3. And again, the literature is very rich, showing that there is a link between the brain omega-3 metabolisms, other link to the diet, but also to age, sex, and stress. As I said before, age is a very important component in the metabolism of omega-3 in the brain, but also in the polymorphism of some genes such as desaturase and elongase that I mentioned before, that are also important for the accretion of this omega-3 in the brain, and mood and cognitive disorders. So to be a bit quick, so there is an inverse relationship in the general population between fish consumption that is rich in omega-3 and the prevalence of depression, but also of cognitive decline in age subject. Decreased level of omega-3 have been described in the blood, but also in the brain of subjects that were diagnosed with depression, but also in healthy age subjects and patients with cognitive impairment. Low level of DHA is associated to cognitive deficit also in infants. And of course, using animal model, it's very easy to modelize this diet. So if you give a diet that is poor in omega-3 to animals, it will be associated to a decreased level of omega-3. And this will be associated also to alteration in some behavior, either social behavior, emotional behavior, but also memory. And one thing that is quite interesting is that, as I said before, those omega-3 are metabolized from the short chain, from vegetable into long chain at the level of the liver, and then they reach the brain, they enter the brain. And yesterday, Bruno talked about that, saying that only a few portions of this long chain reach the brain. And to reach the brain, so there is much debate about how it goes into the brain, but there are specific receptors at the level of the blood-brain barrier. One is MFSD2A, that is a transporter of DHA coupled to a phospholipid. And actually, when this transporter is lacking, it triggers microencephaly, because this transport during development is really important for brain development and the growth of the brain. So, of course, knowing this, many trials have been conducted to give high level of omega-3 to patients with diseases. So the idea was that, okay, if there is not enough omega-3 and that is associated to more diseases or more risk of developing depression or cognitive impairment, then if we give this in the diet as a dietary intervention, it will treat the disease. But of course, it doesn't work that way. And what has been shown is that most of the trials are negative. So giving omega-3 to patients is poorly effective, except in some situations. Some positive effect, and again, Bruno mentioned this yesterday, of dietary intervention with EPA and DHA has been associated to positive effect on depression, especially in combination with antidepressant for DHA and with patients exhibiting inflammation, and this is for EPA. Multimodal intervention with omega-3 improve cognitive decline or MCI in patients only when the level of omega-3 is low, meaning that this supplementation doesn't work in anybody, which is not surprising because we're talking about a nutritional element, so we cannot increase it forever, right? So it's not just because you give it that it will improve everything. So the level of omega-3 before the intervention is very important, as is poorly evaluated. DHA supplementation also has been performed in preterm infants because, as I said before, DHA accumulates in the brain at the end of gestation, and those kids are more at risk to have not enough omega-3. Indeed, many trials have been performed to give more DHA to preterm infants, but the benefits are poor, and actually some benefit have been observed to cognitive development, but not all the time, but the length of gestation is improved. Gray matter sickness is associated to EPA and DHA in age subjects, and a greater hippocampal volume is associated with higher level of DHA and EPA. And increased corticolimbic functional connectivity in depressed youth at risk of bipolar disorder has been also shown. So it's only a few results, but showing that omega-3 can somehow improve some of the symptoms in some situations. And of course, this is linked to the length of the duration of the supplementation, the type of omega-3, the type of subjects, the type of diseases, and so on. And we believe that understanding more how those PUFA are working could help to redesign specific supplementation in specific population. So we get interested in trying to understand how it works. So just to try to explain you, when we started 20 years ago, actually, to work on this PUFA, how it was working was poorly understood, except that those PUFA are esterified at the level of phospholipids. So it's very important to understand that those long-chain fatty acids that have a lot of insaturation goes into phospholipids, and then they will influence membrane fluidity, but also stretching, and influence the functioning of some channels and some receptors. So it influences very much, indeed, lipid-protein interaction and fluidity and stretching. So this has been described especially at the periphery. But another way to use those PUFA is to release them out of the membrane as free forms, and those free forms are metabolized into secondary metabolites such as oxylipins, and again, Bruno mentioned this. Those oxylipins are strong regulators of inflammation. So the one coming from the omega-6, such as arachidonic acid, are more pro-inflammatory, such as prostanoids, prostaglandins, epoxides, while the one coming from DHA and EPA are more anti-inflammatory and pro-resolutive. And this has been clearly shown at the periphery again, where those docosanoids, those derivatives of omega-3 are really strong regulator of the inflammation. So it doesn't counteract inflammation, it helps to resolve inflammation, which is very important because we need to mount inflammation. They can act directly on receptors, and So Wojciech talk about RXR and PPAR that are receptors also for this free form of omega-3, and they are precursors of endocannabinoids, and my group actually showed that it's really important in the regulation of synaptic plasticity, and there are some endocannabinoids derived from omega-3 that are able to regulate synaptic plasticity. And it regulates also the release of some neurotransmitter and the vesicle. So as I said before, arachidonic acid derivatives are pro-inflammatory, while DHA and EPA derivatives are anti-inflammatory, and we are really, we developed a lot of work in order to understand whether this brain PUFA metabolism and the influence of the dietary PUFA could influence oxylipins and the cannabinoids, and also neuroinflammation, especially through the interaction between microglia that we heard a lot about, in particular this morning, because the microglia neuron interaction is very important. This is neuroinflammatory processes that are common to many brain diseases, not only depression, but also Alzheimer's disease and even aging, neuroplasticity, but also behavior. So what I would like to do in 5 minutes, because I guess that I talked too much already, is try to show you some of the results that how we try to understand and to develop research in the lab. So we are playing with diet, actually, either with a diet that is deficient in omega-3 or balanced in omega-3, meaning that the ratio between omega-6 and omega-3 is around the one that is recommended in human. In human, it's 5, and we use here a ratio that is around 6. And those PUFA are only from the precursor form, so coming from vegetables. And then we submit those mice from the first day of gestation until adulthood, and we measure behavior. So here I will talk only about memory. And actually we use specific tests to measure spatial working memory or associative memory. And just here is an example of those mice that undergo a test that is called object location memory, that is characteristic to the measurement of spatial working memory. And as you can see here, the mice that have been fed with a diet that is balanced can perform properly, while this is not the case when they have been submitted to this diet that is deficient in omega-3. And interestingly, this occurs in male and also in female. So both sexes are submitted to this diet. They have some problem in working memory, and they also have a problem in associative memory. So we decided to have a look in the brain. So how does it work? So we focus on the hippocampus and the prefrontal cortex, because we know that in working memory, the connection between these two structures is very important. So actually here what we did is that we put a mice in a maze, and actually one arm is a reward zone with some food, and the mice learn to go in the right arm because they know that there is some food. And then we remove the food and we put the mice again in the maze, and we measure the number of times they go into the right arm, the one that used to have some food in it. And actually what is quite interesting is that when they do that, and they have to choose here at this zone to go on the right or on the left, the synchronization between the activity in the prefrontal cortex and the hippocampus is measurable. So if they do not make mistake, they have this synchronization. And when they do mistake, when they go in the incorrect arm, then there is no more association between the activity between prefrontal cortex and the hippocampus. And what is really interesting is that in the mice that have been fed with a deficient diet, as you can see here, they choose more often the incorrect arm, and they have a desynchronization between the hippocampus and the prefrontal cortex, suggesting that there is really a disconnection between these two structures. And actually, when we looked into the prefrontal cortex of these mice, we could see that the arborization of neurons here in the prefrontal cortex was really strongly altered, suggesting that the neurons didn't grow properly. I remember you that those mice have been submitted to this deficient diet since the first day of gestation, so during development, even if they are adult. So the prefrontal cortex has some the neurons are not in good shape, I would say, suggesting that the connection could be impaired. And we also did some measurement in the prefrontal cortex and hippocampus of mRNA profile, so using not single cell, but RNA-seq. And what we found is that there is an upregulation of some genes that are more involved in cell communication, but also immune system and brain development, not an upregulation, sorry, dysregulation in the expression of those genes, both in the hippocampus and in the prefrontal cortex, suggesting that something happened really early in life and that the exposition of this deficient diet could really involve a dysregulation really early in life. So we decided to go for young animals, and so we submitted them to the two diets, the balanced diet or the deficient diet, but then we studied their brain and their behavior and everything at weaning, so really early at the end of the lactation. And what we could find is that those mice, when they are submitted to a working memory task, again, they have an impairment. So if they have been fed with the omega-3 deficient diet, at weaning, they're not able to recognize the right arm, so they have a spatial memory impairment. And this was accompanied by an alteration in synaptic plasticity, here measured as long-term potentiation. And you can see here that after a stimulation, electric stimulation, there is no response in the brain of this deficient mice. This is the opposite, sorry for the color. And again, we measured the dendritic arborization in the hippocampus, and we could see that there is a decrease in the dendritic arborization of the mice that have been submitted to the diet deficient, but also a change in the number of spines at the level of the CA1 area of the hippocampus. So what we know is that the number of spines grow during development, and that those spines have to be removed specifically because not all the spines are useful. So there is a kind of exuberant production of spines during development, and then they are removed specifically, and this is called synaptic pruning. And synaptic pruning involves several mechanisms, but one important cell to do that is microglia. So microglia are phagocytic cells, and actually they are able to recognize specifically the spines that are not useful, and they remove it through eating kind of those spines. So we decided to have a look to those microglia to understand whether the deficient diet impaired microglia, and that maybe those microglia were hyperphagocytic and were eating more spines, and that was why at weaning those neurons had less spines. So what we did is that we took out the microglia of the brain of these young pups. We measured some specific genes, and we could find that the microglia have an impairment in the homeostatic signature, suggesting that those microglia are not mature anymore, they are not at they don't have the right profile, and they are more like embryonic microglia, and those embryonic microglia are really well known to phagocyte a lot spines and cells. The other thing that we did is that we measured the profile of PUFA in those cells, but also of oxylipins, because as I said before, omega-3 can influence the oxylipin profile, and this was the case. And what we found is that in microglia of these mice that have been fed with a diet that is poor in omega-3, there is more arachidonic acid, that is an omega-3, and more of these oxylipins derived from arachidonic acid, while DHA, EPA, and their derivatives were decreased in microglia. And actually, when we looked at the synaptic pruning through the measurement of synaptic inclusion in microglia, we could see that there is an increase in the spine inclusion in microglia, suggesting that microglia really had phagocytes more spines in the brain, in the hippocampus of the mice that are deficient in omega-3, and we confirmed it by using other approaches. So we were wondering whether the oxylipins that were upregulated in microglia and that are known to regulate inflammation were involved in this hyperphagocytic activity of microglia. So we submitted microglia in culture to some of the compounds that we measured that were increased in microglia, such as the HETE, the 12, the 8, and there are many others, but I will show you only result on this 12. And actually, 12-HTE is able to upregulate phagocytic activity of microglia. And when we block the production of 12-HTT by using a flavonol inhibitor that is called bicalene, that is able to block the production of 12-HTT, this hyperphagocytic activity of microglia is reduced, suggesting again that the omega-3 deficient diet just polarized microglia into hyperphagocytic microglia, and that 12-HTT that is produced is involved in this hyperphagocytic activity. And if we block this in vivo, this activity, we can improve the memory alteration that was found in those mice that have been fed with this omega-3 deficient diet. Again, suggesting that the way microglia and neurons interact in those brain is really involved in the memory impairment that we measured. Il me reste quoi? 1 minute, 2 minutes, 0 secondes? More or less, okay. So this really early life interaction between microglia and neuron is very important. So one question is, can we reverse this? So if mice have been submitted to a diet that is deficient in omega-3, they have this memory impairment, they have this dysregulation. So can we, if we give back omega-3 after weaning, can we change this? Is it possible to reactivate everything? So this is what we did here. So again, we use mice that have been submitted to a balanced diet or to a deficient diet. And the third group, mice were submitted to a deficient diet until weaning and then back to a diet that is balanced in omega-3. And we measure memory in male and female. And actually, the interesting thing is that male and female, when they have been fed with a deficient diet, they have the memory deficit. And when they have been switched to the balanced diet at weaning, and memory was measured at adulthood, only female can recover. It is not the case for male. And actually, the LTP is improved in both male and female. The DHA level is improved in male and female, but not arachidonic acid. As you can see here, so there is an increase in the brain of those mice that have been fed with a deficient diet of arachidonic acid, both in male and female. And when they are switched to the balanced diet after weaning, arachidonic acid goes back only in the brain of female and not of male. So we believe that this is associated to a different profile in oxylipins, and this is the case actually. So male and female have a different profile. And we also believe that this improvement in DHA, but not in arachidonic acid, is involved in the spine formation. And as you can see here, only female, when they are switched to the good diet, have an improvement in spine density in the hippocampus. And when we measure, again, the gene that are differentially expressed in the hippocampus of this animal, we could find that male have an improvement in the phospholipid and fatty acid metabolism, while female have an improvement in the genes involved in synapse organization and dendritic spine organization. So we are not trying to understand whether microglia are involved in this and whether the fact to give back omega-3 to female can improve this, but this is ongoing. And this is a summary of all the data that I've shown you, and I would like to close here by thanking all our collaborators, but also the talented people that contribute to this work, and they are here, and really they did a wonderful job, and also our financial support. And I would like to thank you for your attention.
Thank you very much.
Thank you, and I'd like to start by thanking Dr. Leboyer for the invitation to be here and for you to stay until the late hour to see my presentation today. So we're going to actually talk a little bit more about ketosis, not so much about ketogenic diet. And the reason for that is because the mechanisms behind is actually on the ketones, and how we get to ketosis in our bodies comes from different ways. So I thought that I would start by trying to just show you my disclosures, but to answer why should we study ketosis in psychiatry. Is this something important? Should we actually pay attention? And I think the answer to this question is actually quite simple. Energy demand in our patients is increased. So what do I mean about energy demand being increased in the patients with psychiatric disorders? And I hope through this, hopefully it will take less than 20 minutes, to convince you that actually patients with psychiatric disease have demands in the brain, in the energy processes that can be restored via our nutritional approaches, and one of them is nutritional ketosis. Let me see if, oh, perfect, it's working. So I'm going to show you a little video that Mitochondrial Innovation Initiative, which is a foundation at the University of Toronto, that I'm the director for that. It's been putting some awareness of how this energy metabolism is actually connected with the brain. Energy in our cells is produced by an organelle that I hope you all know, that is called mitochondria, and is my favorite organelle in our body, because without mitochondria, we don't have energy. If we don't have energy, we simply don't have life. So hopefully you enjoy this 1-minute video.
The human body needs a reliable supply of energy to power our muscles, organs, and especially our thoughts. The brain uses more energy than any other organ, using 20% of the body's total energy to power brain function and neuronal signaling. Energy is generated by mitochondria, which are the powerhouses of our cells. Impaired mitochondrial function leads to major cellular changes. This is because they are much more than powerhouses. They are important hubs that regulate critical biochemical pathways involved in tasks like memory and learning. Brain synapses are the way that our brain cells communicate with each other. They require high mitochondrial density for proper communication, also called neurotransmission. If mitochondria are not working properly, the communication of brain cells will become impaired. This can lead to changes that will impact several functions in your brain. For example, you might not have enough energy to deliver neurotransmitters to brain regions involved in maintaining your mood or behavior. Researchers are working hard to understand this process. A growing body of evidence links mitochondrial dysfunction to complex brain diseases such as Alzheimer's, Parkinson's, bipolar disorder, and many others. Further investigation of the links between mitochondrial dysfunction and brain disease will help to develop a mitochondrial diagnostic tool to identify patients with complex brain disease who would benefit from mitochondrial therapies. Faster diagnosis and efficient treatment will ultimately improve the quality of life for patients. Mitochondria research matters to millions. Join us and spread awareness.
Okay, we now can go and enjoy Paris. The lecture is over. Oh, sorry, how do I get out now over here? Oh. There. Okay, so jokes aside, I think the video actually touched a little bit on the importance of our energy metabolism for the brain function. And this slide is just to remind us how complex it is. We just heard about Sophie, the lipids, and how complex it is in the brain. And lipids are actually essential for maintaining mitochondria healthy and functioning. So our talks are quite interlinked. In a normal processing brain, it's very tight and is also... Oh, this is interesting. Oh my goodness. Okay, I guess I will get in track by the end of the presentation. So the brain is very... In a normal healthy brain, we have enormous amount of connections between different types of neurons that is happening at every single moment. The glutamatergic neurons conversing with the GABAergic system, with the dopaminergic system, the serotonergic system, and nowadays we're learning more and more and more about the endocannabinoid systems in our brain. In a normal functioning brain, we need to have mitochondria releasing quite fast energy, or ATP, adenosine triphosphate, for the synapses to release calcium and to actually open the vesicles to release neurotransmission. And this has to happen in milliseconds and every single processing time in our brain. Now imagine the patients that you treat with psychiatric disease, which already have a disconnective on this system, which are working harder and harder and harder to maintain the balance with this system. What do you think is their energy demand? Lower or higher? Higher. Thank you. Because that's what I meant about the first phase. They are already working in this high demand for energy to supply them necessity of maintaining this neurotransmission, the connectivity between each of this system, this complex system, working properly. But patients with bipolar disorder, with schizophrenia, with Alzheimer's, with Parkinson's are known to actually not have a well-functioning mitochondrial system. We're not going to enter today on all the details of how this system is actually not working properly in different diseases, but I would like to walk you through about some of the mitochondrial metabolic pathways that are actually dysfunctional in some of patients with psychiatric disease. You could ask myself, is all the patients with psychiatric disease that actually have mitochondrial dysfunction? The answer potentially is no. Do we know how many % of the patients actually have mitochondrial dysfunction in all the diseases? No, we do not have a great answer for that. From the literature, from clinical studies, and more recently coming from light epidemiological studies, in bipolar disorder, we now know that an expectation is between 20 to 25% of patients with bipolar disorder do have dysfunction in mitochondria, dysfunctional mitochondria. And this is mostly related to the data that is coming to light on metabolic syndrome, related to the data on diabetes, and also on lactate. And that's what we're going to talk right now. When we look to this system, you probably have seen this from your biology courses. You eat, you get glucose, but the glucose has to be metabolized, and they will be entered into the acid, the citric acid or the Krebs cycle to actually go into the oxidative phosphorylation and produce ATP, right? That's the basic biochemistry that we all learned at one point. Hopefully you didn't forget it. So the glucose has to be converted into pyruvate. And the pyruvate is the driver on this process that will take inside of the mitochondria into the pyruvate dehydrogenase and start into the acetyl-CoA to drive this process of the TCA cycle. When we talk about breaking down, I love to think about it's like baking. If you put it like a glucose and you put in too much sugar on your cake, no, it will get heavy. But if you put too little, it will not work. So the right amount is necessary. You know, anytime we say do not eat sugar, we have to remember that glucose can be broken down from other forms of food. So it's not because you're just not eating the sweets, you're not going to get glucose in your body. You will actually get other forms that can be converted in biochemical assessments, because it's important that we remember that glucose is essential. However, the amount that we eat is that what we need to be caring for. So when we change these glucose levels in patients with psychiatric conditions, and I show you here a data from Professor Leboyer and Dr. Ophelia Godin, who have demonstrated that here in France, for example, the levels of metabolic syndrome, which are characterized in 5 criteria: high waist circumference, hypertriglyceridemia, low HDL, high blood pressure, high fasting glucose concentration. If we look in bipolar disorder, it's about 20%. In depression, resistant depression, about 38%. Glycemia, the high glycemia in non-treated patients is 72%. That is incredible data. And why this is actually something that could be important for metabolism. When you have high levels of glucose and you have insulin resistance, you increase a process called glycogenesis. Glycogenesis, if you remember that picture that was just in a slide before where you had glucose and you had fatty acids, this glycogenesis is taking the fat and actually converting back in glucose. Because when we have this insulin resistance, we have high levels of glucose all the time in our body, we teach our cells to actually go into the inverse, to say, give me more glucose. And that becomes a vicious cycle because all the side in this slide on the right-hand side, it will actually get diminished. So you can't process oxalate. You can't get into the acetyl-CoA. Your TCA cycle doesn't go down. So the cells are now deprived of energy. They don't have enough amount of ATP necessary to go through. And then what the body does to support the system, it's very smart. It actually accumulates lactate. It produces another way of producing the ATP for you to not starve and not die. So that's a survival mechanism. The cells actually do it at control. And if you go into patients with psychiatric conditions, and here I show you data from my own research on bipolar disorder, lactate levels are consistently increased in patients with bipolar disorder. And that's true for schizophrenia and true for many other conditions of psychiatric conditions. You will see this in the periphery and in the brain. So levels of lactate have been replicated over 8 times in patients with bipolar disorder via MRS, magnetic resonance spectroscopy, and over 12 different times in the periphery of this patient. So it's a result that is really consistent. So if we think now, okay, patients with bipolar disorder or other psychiatric conditions have this insulin resistant, that this hyperglycemia, pushing my system to drive my system to anaerobes, is accumulating lactate. Is the accumulation lactate good or bad for the body? Bad. Well, yes and no. Short term, maybe it could be used as a signaling messaging. It could be used as actually an energy production message. When you run, your muscles produce lactate, and that's okay. You actually needing that glycolysis to go fast. You lose fast, it's fine. The long-term in the brain, the long-term accumulation of lactate shuts down other signalings, especially the regulation between dopamine and GABA systems. And that is extremely important for patients with psychiatric conditions. And that's an evolving science with no exact answer yet. If you go and you start looking at the literature, you will definitely find different results that are controversial, but I think we are getting much closer and closer over the years to understand better. The final part of the cycle is also showed here by data on my research in bipolar disorder, which demonstrated these patients in the peripheral, in the post-mortem brain prefrontal cortex. We replicated in 3 different sets of samples from 3 different brain banks and have low levels of and a complex 1 enzymes, including the NDUF7. So this means that your oxidative phosphorylation is actually not producing enough flow through the 5 complexes, so the levels of ATP in the end potentially could be impaired. We also did an interesting work with a group in Sweden with Michael London, and which his data demonstrated increased levels of isocitrate, which is one of the within the pathways of the citric acid, so not producing enough NADH. Well, let's go back to the question. Why should we study ketosis? Well, again, why do you think maybe ketones could be good? Ketones, in this case, in the scenario of mitochondrial dysfunction, they are actually another set of energy productions for your cells. They help your body to actually achieve the production of ATP that is necessary, that you're not getting through because your mitochondria is being impaired through the oxidative phosphorylation and is being pushed into anaerobiosis instead aerobic formation. Also, the different types of ketones, which are the acetoacetate and the beta-hydroxybutyrate, especially the beta-hydroxybutyrate, it acts as a HDAC, which means acetone deacetylase class 2 inhibitor. And that is interesting because it modulates the production of antioxidant genes, which will help to decrease the reactive oxygen species in the system. At the same time, Marion, you're going to like this one, BHB is actually an anti-inflammatory as well, so it modulates inflammation via the NLRP3 inflammasome. Topic for another day. But here, focusing on the energy metabolism, if we look at this graph here, is showing that from the periphery is actually these ketones can now be derived to our body. And why this is so important? Because it crossed the blood-brain barrier, and so many years we have been studying mitochondria in my career and trying to focus on finding molecules that we can actually drive and cross the blood-brain barrier. So nutritional ketosis has actually an interesting aspect because we're treating patients with a molecule that is endogenous of your body that actually can cross the blood-brain barrier and potentially have the effects that we hope it will have in these patients. Cautionary point here. If you are trying to enter any of diets that actually change ketosis level, ketone levels, please talk to your healthcare professional. And if you are a physician work with a nutritionist to help you to guide through this process. Has many different ways that your body can enter in ketosis, and that can be via intermediate fasting associated with a vegetarian diet. It can be with a ketogenic diet. There are so many types of a ketogenic diet. And there are also supplementations that actually can help your body to go through. This will be a 3 hours lecture on its own, so I will not enter in details, but I will urge you, if that's something that you are interested in learning more, please enter in the website Metabolic Mind. They have enormous resources on where to find resources in these 3 topics of how to induce ketosis safely. And the patients always should be doing this accompanied by a healthcare professional, never alone. There are consequences to your body that you should not do. If you have diabetes, for example, you can enter in ketoacidosis, and you don't want to actually get to that point. So it's a treatment. We're not talking here about a change in a lifestyle. When I'm talking about treatment of ketosis, is actually a treatment that changes your cellular biochemistry. So it should be treated as a drug, not as a lifestyle change. Ketones induce neurotransmissions, and that's what I was talking before, and that's a very heavy topic right now in my lab studying, where we are really focusing on the beta-hydroxybutyrate effects on the HDAC inhibitor that leads increase of gene expression involved in redox balance and in promoting mitochondrial function. It also balances the synthesis of release of GABA and decreases dopamine. The WHO, I'm not going to enter on all the pathways and details today because it's unpublished yet, but it's coming out very soon. It leads to an increase on the pathways that releases GABA and that promotes a decrease on dopamine. And as you know, this is actually quite important for patients with bipolar disorder. How we do that in a lab, we actually use white blood cells from patients, transform very slowly on a very long process to transform with iPSC. We do not use viral transfection because in our lab we have tested both viral transfection and episomal vectors and noted that when you use viral transfection, it changes mitochondrial metabolism, so that's why we avoid that type of work for working with mitochondrial metabolomics and mitochondrial analysis in iPSC cells. Once we get to iPSC cells, we transform that into brain organoids. In our case, we are using 4 brain organoids, which are enriched in dopamine, and then midbrain organoids, and then we combine both of them forming assembloids, and these assembloids then now have the 2 systems that we want to be able to study and use. From that systems, we noted that patients with bipolar disorder have a lower level of OXPHOS, so decreased ATP and decreased levels of complex 1, and they have a ratio to green to red that is more green, meaning their mitochondrial membrane potential is depolarized, and that is reversed by some of the treatments that we have been testing, and the results are coming out to publication soon. But just as a teaser, what we actually have been noting is that the beta-hydroxybutyrate is more effective than the acetoacetate into promoting the mitochondria functioning in these assembloids of patients with a bipolar disorder. There are also, this is the website that I want to leave with you today, great resource on the area of ketogenic diet. So if that's of your interest, please do take a look on it. There are several trials and several clinical cases that have come to light in the past years with effectiveness of ketogenic diet for patients with bipolar disorder. This is not exclusive at all of bipolar disorder. We have seen now trials with effectiveness on OCD, with anxiety, Alzheimer's, and I believe the last one that I saw, but is not a complete trial yet, was Parkinson's. So there is a vast range, and the type of diets that they use is modified ketogenic diet, is not the traditional ketogenic diet. So that is very strict and heavy. They actually allowed about 30 to 40% of carbohydrates in this diet, so it's kind of going into more a low carbohydrate diet. There is actually 2 trials which I thought was very interesting. They are ongoing, they don't have data yet, but they are actually using either pescatarian or vegetarian ways to induce ketosis, which I think it is a fantastic and interesting way, because as we all know, with the processed red meats, there are other studies that have shown to be not beneficial for the brain. So interesting studies coming up here. Some results have been persistent over long term. I always get this question, do you change lipid metabolism in the patients? Surprisingly, actually, the patients had been stabilized with their lipids. We have not seen to become that these patients have a high levels of cholesterol. There was one patient in one of the trials that actually demonstrated that, but we then was discontinued, of course, from the trial. But so far, that's a safe part of the diet. There is fatigue, constipation, drowsiness, hunger, thirsty. It's a very common one. But yeah, the other ones are less common. But the most common one so far has been fatigue. on these patients. And I will skip this one in light of time. Invite you to visit this website, the Mitochondrial Innovation Initiative. If you is a lover of mitochondria as I am, this is a society for you. And it brings together from patients to researchers and clinicians. We all work together to really transform the understanding of mitochondrial health and hopefully bring this one day to everyday medicine. We should be finding new treatments that target this area. There is lots of complex questions to be answered, and I am so grateful to have a phenomenal team working with me, and I'm very thankful for being able to be here working with Dr. Leboyer and Dr. Godin has helped me incredibly during this journey, and thank you for the Bazuki Research Fund for funding this research. Thank you, everyone.